We're moving on to the part of the unit where we're going to talk about each of our senses. So we've talked about sensation in general, and now we're going to get a little bit more specific. It's important to understand that all of the senses operate much in the same way. They're taking in some form of energy from our environment. At the sensory receptors, transduction is occurring, where that energy is turned into action potentials. But the difference is each type of neural message is going to be processed in a different part of the brain depending upon which sensory receptor is receiving the information. So, for example, light energy transduced is going to go to the visual cortex and the occipital lobe. Sound waves that enter through the ear and are transduced into neural messages are going to be processed by the auditory cortex and the temporal lobe. So the difference really comes in the region of the brain where that particular type of information is processed, and that's what creates all the different types of sensation that we experience. If you remember from a few videos ago, we talked about the fact that we use light energy to see. Of the entire electromagnetic spectrum, there's only actually a very tiny part of it that is visible to humans, or that is detectable by the human eye. And we'll talk about that a little bit more, about the different wavelengths that we're able to see when we discuss color vision in our next video. Here's a little thing that you could do to find your dominant eye. I'll play this real quickly and you can try it.
To find your dominant eye, create a triangle using your thumbs and forefingers as shown here. Now frame a distant object, such as a clock on a wall, within this triangle, keeping both your eyes open. Then close your left eye. If the object stays centered, your right eye is your dominant eye. If the object moves out of center, your left eye is your dominant eye.
So that's kind of neat. Now you've been able to figure out which is your dominant eye. Our first goal of today is to talk about the structure of the eye. So we do not need to know all of these various structures, okay? Most important to us are the cornea, which is this outer layer, the iris. You already know your iris as being the part of the eye that is colored. So your iris is actually a muscle that controls your pupil. The lens, your lens, and the retina. The retina, I like to say, this back part of the eye is where the magic happens, because this is the part of the eye that contains photoreceptors, the rods and the cones that are responsible for transducing incoming light energy into action potentials. Those action potentials are going, that electrochemical signal, the neural messages, are shuttled out from the retina through bundles of nerves that make up the optic nerve. And the optic nerve is what we're going to see carries that signal all the way back to the visual cortex, where vision is processed. Just a little bit for those of us that wear glasses or some type of contact lens, corrective lenses, myopia or nearsightedness occurs when the eyeball is too long. This is what I have. So you see that the image is projected just before the retina instead of on the retina, like in someone who has normal vision. Individuals who are farsighted, like my daughter, you can see things far away but have difficulty seeing things up close. It's because the eyeball is a bit short, and the image, instead of being projected right onto the retina, is projected actually behind the retina. For those of us with astigmatism, it means that our cornea is oval shaped, and that results in two focal points, which makes our vision blurry in some cases. I know that in my contacts, at least the early contacts that I wore, they actually had a weight on them, like an undetectable weight, but a weight nonetheless, that would always keep the lens in the same position on my eye so that it could correct for my astigmatism. So how vision works. Now this is the part in your notebook where you have a list of anatomical structures, and it's important that you have a good overview into just the general function of each part. I do not want you copying the slide word for word. Please make sure that you put things into your own words. It might be a good idea to pause the video, write, and then listen. So light enters the eye through the cornea, and the function of the cornea is to bend light and to provide focus. The iris, the part of your eye that's a color, mine are brown. Some of you might have blue eyes or green eyes, or my dad actually has a brown eye and a green eye. He has two different color irises. But the iris is the muscle that adjusts the size of the pupil. The pupil is the hole in the center of the eye that allows light to come in to strike the retina. The lens is responsible for focusing those incoming light rays on the retina. And it accommodates, meaning the lens will change shape depending on if an object is near or far away. An upside down image of what we're looking at is formed on our retina. The pupil reacts in different ways to varying amounts of light. This is what our pupil would look like in normal light. Pupil size reduces in bright light. So if you were to shine a flashlight in someone's eyes, the iris muscles would contract, and that would result in a very, very tiny pupil. In dim light, the iris expands. Okay? So what is happening when you go to the eye doctor? At least for me, because I wear contact lenses, they have to dilate my pupils every time I go with eye drops. And what that means is that I leave there with my pupils looking something like this, but they need to really get a nice view of my retina when they're doing the retinal exam. So now we're honing in on the retina, and I just want to quickly skip to this slide, and then I'm going to go back, just so you could see what the retina looks like. So this is the back of the eye. If we were to look at the eyeball being like this is the back of the eye. When we're referring to the rods and the cones are these cone-shaped objects here, which make up the center of the retina. As you get into the peripheral vision, you see more of these rod-shaped objects, and these are called rods. Collectively, the rods and the cones are known as photoreceptor cells. We'll come back to this slide in a second. So photoreceptors are these specialized neurons, the rods and the cones, on the back of the eye, which is called the retina. Again, where the magic happens, because these photoreceptor cells receive or take in light energy and respond by creating neural impulses, which just means they take in light energy and they fire action potentials. Cones and rods have very different distinctions. So cones allow us to see color. They are concentrated in the fovea, which is our focal point. So I am focusing right now on this screen. It is very crisp, it's very clear, because my cones or my fovea is focused on it. Everything in my peripheral vision is a little bit foggy, and we're going to learn even lacks color vision because rods are in our peripheral vision. Rods work better in low intensities of light. Like if I'm navigating my room at night and it's dark, the light's very low, I don't see color because my rods are at work and not my cones. My cones need bright light in order to be activated. So this is a nice little chart that kind of summarize the differences between the rods and the cones. Cones concentrated in the focal point or the fovea allow us to see in color, fine detail. They're less sensitive to low light. They're used for our central and our detailed vision, whereas our rods, we don't see in good detail. They're concentrated in our peripheral vision. They only allow us to see black and white. They are much more numerous than the cones. They're sensitive in low light, meaning it's what we're using to navigate around a space in the dark. They're used for peripheral and in night vision. I like to remember cones, color. Really easy way to remember the cones, color. So coming back here to our picture of the retina again, this is our fovea. The fovea is a cluster of cones that make up the focal point of our vision and allow us to see in very fine detail. Here's another image of the retina. Again, just like we were on the left side, we're looking at the back of the eye or the retina. But this is showing you really nicely how light comes in, activates the rods and the cones, and the signals actually travel this way. Action potentials travel this way to the bipolar cells and the ganglion cells. The axons of the ganglion cells make up the optic nerve. Really interesting point. The area or the point at which the optic nerve exits the eye contains no rods or cones, and therefore we all have a blind spot. And we are going to learn in class how we can find our blind spot. So now we're looking at a dorsal view of the brain. We're looking at it from overhead. Here are our eye sockets. We learned a little bit in our biopsych unit. We learned a little bit about how vision is processed in a contralateral way. So information from the left visual field is processed by the right hemisphere and vice versa. And that is because visual information, the optic nerve itself, crosses at what is called the optic chiasm. This is what allows for that contralateral processing of vision. The part of the nucleus that receives these action potentials or these signals and transfers them to the visual cortex and the occipital lobe is the LGN of the thalamus. And we already know that that's the function of the thalamus, to take in sensory information and to shuttle it back to the part of the brain where that particular sensory information is processed. So from the eye along the optic nerve to the LGN of the thalamus back to the primary visual cortex in the occipital lobe is the pathway for vision in the brain. We've talked about sensation, and we've also given a little bit of an overview of perception. So we know enough to recognize that our brain has to interpret information that's in front of us. And in the occipital lobe, we have these cells called feature detector cells. These cells are located in our visual cortex, and they're able to detect specific features of a stimulus, like its slant, its angle, its boundary, its color, its shape, movement, location, its length. And what that allows for is for us to recognize what the object is. It allows us to identify objects. If you think to the back to the waterfall effect, that was referring to the activity of movement feature detector cells. There are certain cells that specifically detect left-right movement and others that detect right-left movement. So in order to identify an object like this, think about what our feature detector cells would need to detect. The color, the shape, the boundary, the angle, the slant, in order for us to recognize this as a rose. And that's the job of the feature detector cells. It's important to realize that the brain is always engaging in what's called parallel processing. So the brain is processing so many different kinds of information at the same time. If I'm looking at a scene, we have the retinal processing, which involves the rods and the cones, the bipolar and the ganglion cells. We have that information being sent to the visual cortex, where the feature detector cells are going to respond to the different features and allow us to identify what the object is. This identification is occurring at higher levels of the brain, and it's also drawing on areas that are involved in storing memory that allow us to kind of complete that recognition task so we know exactly what it is that we're looking at. But the bottom line is that this is all happening instantaneously. The brain is processing all different types of information at once, and this will finish us off here. There are two different pathways where visual information is sent. If you think back to blind sight and the little video that we talked about when we discussed consciousness, this is kind of reflective of that phenomenon of blind sight. Visual information doesn't get sent only to the visual cortex. It's also sent to what are called the what and the where pathways in the brain. The temporal lobe, or the what pathway, allows us to identify what the object is that we're looking at, whereas the where pathway in the parietal lobe is allowing us to locate where that object is in space. So again, yes, we have the visual pathway that we've learned about that sends those neural signals back to the visual cortex, but we also have visual information going to other parts of the brain that also enable our visual processing.